I do not have a share button here, Ryan. There it is. Yeah, got it.
You see it there?
I have it, yes.
Very good. Well, thank you for joining us in our session on the U.S. Celiprolol trial for VEDS. An update from Dr. Adrian Quartel. So thank you for joining us, Dr. Quartel. Adrian Quartel is the Chief Medical Officer of Zevra Therapeutics, formerly Acer Therapeutics. Joining us today as well is Dr. Xavier Junemet, who will be involved in some discussion here towards the end of the talk as well. And as you know, if you have any questions, please feel free to submit those via the Q&A function of Zoom, and we'll be monitoring the chat and the Q&A box for any questions that come in, and we will ask those of the doctors after the presentation is over. So welcome, Dr. Quartel.
Thank you, Ryan, for the introduction, and thank you everyone for listening in. Also welcome Dr. Junemet, who gave a very good presentation an hour ago in regards to the use of angiotensin receptor blockers, or irbesartan, for patients with vascular Ehlers-Danlos syndrome. Dr. Junemet has agreed to join me because these are fairly similar trials, and in the Q&A session, if there are very specific questions that cover both of the trials, I thought it would be the best unbiased way to answer these questions if Dr. Junemet would provide some input in that as well. As said, my name is Adrian Quartel. I'm the Chief Medical Officer of Zevra Therapeutics, and Zevra Therapeutics is probably a company that no one of you have heard of. As you may be aware, the Discover trial was actually started by another company called Acer Therapeutics, and I also used to be the Chief Medical Officer at Acer Therapeutics. Acer Therapeutics, although did great work, ran into some financial problems and was acquired by Zevra Therapeutics in November 2023. And as part of the acquisition, I moved over as a Chief Medical Officer from Acer Therapeutics to Zevra Therapeutics. Just very briefly about Zevra, it's a company focusing specifically on rare diseases, and their mission really is to bring life-changing therapeutics to people living with rare diseases. Small company, but an experienced team, specifically when it comes to rare diseases. Commercial excellence, we have one approved product for a rare metabolic disorder, and a growing pipeline specifically in the rare disease space. Different stripes, same animal. It's curious. So Zevra Therapeutics is from the Greek zevra, which is this zebra, and our logo is the zebra. And as everybody knows from the Ehlers-Danlos space, the Ehlers-Danlos Society logo is also a zebra. So there's a little bit of overlap that is, serendipitous, but interesting nonetheless. I'm going to speak today not about VEDS. Obviously, everybody that dialed in is aware of VEDS, so I'm going to focus mainly on Celiprolol, the drug that we are testing. A little bit about the history of Celiprolol in the treatment of VEDS, and talk a little bit about the VEDS study that was performed 10-15 years ago in France and Belgium. Specifically because that really was the basis for the design of the Discover trial. And that's the trial that's currently ongoing to test Celiprolol in the treatment of patients with VEDS. Then we'll talk a little bit about the Discover trial itself as well. So with that, obviously, the disease overview: autosomal dominant connective tissue disorder caused by mutations in the COL3A1 gene, specifically for collagen synthesis, and characterized by arterial aneurysms, dissections, and ruptures in the arteries or hollow organs. And this is obviously a devastating disease. You don't know when the event may occur, and these events are quite often life-changing or even worse. With no approved therapeutic option, no medication that is specifically approved for VEDS, there is a significant unmet need. We all know that there is a lot of off-label prescription of medications, but no drug has been specifically tested to show that it makes a significant impact on patients with VEDS, up to a point that a regulatory agency would agree to the approval for that drug for that specific indication. The VEDS study did a good job at getting there. And that VEDS data was actually used for a filing for an approval in the United States about 5 years ago. And the FDA did review all the data from the VEDS study and decided that the data was interesting, was definitely directionally positive, but did not meet the minimum criteria for an approval. And subsequently, the FDA refused to approve the drug and requested an additional trial, and that's how we got to the Discover trial. As said, we have good efficacy data from the VEDS trial showing a significant reduction in risk of arterial events in patients with not only the COL 301, but even if you use looser criteria, the Villefranche criteria, I'll get into that a little bit later, you see a statistically significant, more importantly, a clinically meaningful improvement in the time to event and the number of events in patients treated with celiprolol compared to those treated with placebo. There's a long-term observational study, and there is additional data from a cohort in Sweden that all supports the use of celiprolol. And based on that, in countries where celiprolol is approved, and celiprolol was approved in the mid-1980s to the late 1980s in most countries, except for the United States, for the treatment of hypertension or other specific cardiovascular disorders. As such, it is available, and physicians are allowed to prescribe this off-label. And since it's been approved for such a long time, they're considered generic drugs, so they're relatively cheap to get. So based on that, we decided to run the Discover trial, which is a phase 3 decentralized pivotal trial, to assess the effect of celiprolol on patients with VEDS. Why celiprolol? A lot of people keep on calling it a beta blocker. It is a beta blocker, but I always call it an adrenoceptor modulator because it is not a straight beta blocker. And the reason for that is it does not just have beta-1 antagonism. It actually has what's known as intrinsic symptomatic activity, which it's not only an antagonist, but it also can be a partial agonist, which is feels a bit odd to say that, but it can do both on the beta-1. So it is not a straight beta-1 inhibitor. Beta-1 inhibition or antagonism causes a decrease in blood pressure and a decrease in heart rate. And what we do know in patients that have VEDS, most of these patients are normotensive, meaning they have a normal blood pressure, they are not hypertensive, and also usually have a normal heart rate, so they don't have tachycardia. So in those patients, celiprolol actually does not have a significant effect. It does not significantly lower the blood pressure or the heart rate. So in that sense, it is an odd one out in the field of beta blockers. It has alpha-2 antagonism, which causes vasodilation, which is something that you would like to see in a disorder where ruptures in large arteries do occur. There's beta-2 agonist, which causes the relaxation of hollow organs. It's cardioselective, meaning that it has, as I said earlier, intrinsic symptomatic activity. So acts both as a beta-1 antagonist and as a partial agonist. And there's some hypothetical data in the background, and this is not proven yet, and additional research needs to be done, is that it may upregulate TGF-beta, which is responsible for or is involved in the extracellular matrix layout for arteries, and therefore may have a beneficial effect as to how collagen 3 and collagen 1 are part of a appropriate extracellular matrix. I said beta or VEDS patients are normotensive, so the beta-1 antagonism has not shown to have any clinical benefit in VEDS patients. I will go into that a little bit later. There is no difference in the blood pressure in the patient that received celiprolol in the VEDS study versus those patients that did not receive. So these are patients that were followed for up to 5 years, and they did not see a clinically meaningful or statistically significant difference in blood pressure in the patients that received celiprolol versus placebo. And then we have some data that I will show to you later, which is claims data. So this is not clinical trial data. This is data that you derive from data sets that you can acquire from insurance companies to see if a patient is using a drug, what kind of side effects can you actually correlate with that. And based on that, you can make an assessment that, again, is not proven scientifically, but it can make an assessment that there is a correlation or no correlation between the use of a drug and a certain event and that could guide you into doing further research. And that certainly guided us in doing further research with Celiprolol. Celiprolol is the only drug to be thoroughly studied. Now, this is a statement that is getting a bit old, and obviously there's a lot of ongoing research, and certainly angiotensin receptor blockers are currently being studied in a variety of ongoing investigator-initiated trials, certain academic centers, and there's also a little bit of published data in regards to that. But specifically studied in a large cohort of patients, probably Celiprolol, I would argue, is the most studied one. And if approved, we will be the first drug to be approved specifically for the treatment of vascular Ehlers-Danlos syndrome. As said, this is specifically studied for the United States with the sole purpose to get Celiprolol approved in the United States. I said it is not available at all in the United States for treatment unless you use it through parallel import, which technically is not legal. But luckily, a lot of patients do find ways to get it in their hands and are able to treat themselves with it. Mechanism of action, I'm not going to go into this too much. I just want to emphasize that Celiprolol is a selective adrenoceptor modulator. It is not a specific beta blocker. Here's the list of the most common beta blockers, and as you see, they all have very specific part as to how they actually work. Within this setup, Celiprolol has alpha antagonism. It is beta 1 selective. It has positive ISA, and therefore it has its own place within the treatment paradigm. And as far as we are concerned, and as far as we have data, it cannot be swapped out for any other beta blocker to treat VEDS, or there's limited data available to suggest that. So claims data to support that, and this is claims data that is probably from 2018, so it's 6 years old. And what you do is you basically look at a very specific disease, in this case, vascular Ehlers-Danlos, and you look at what treatments are prescribed for these patients. So you download a database from a health system. You can do that from the NHS in the United Kingdom, or you can do that in the United States through a claims database that is available. And what you do is you say, okay, for all patients that have a diagnosis of VEDS, what treatments are they on? And then you can look at no antihypertensive therapy, or if there are antihypertensive therapy, what are they on? Is it any, or is it specifically a beta blocker, an ACE inhibitor, an angiotensin receptor blocker, or a calcium channel blocker? And then you look for that specific population. How many events did occur over the time that they were on that treatment? And these events are specific for vascular Ehlers-Danlos events. So these are cardiovascular events such as a dissection, rupture, or aneurysm in a artery, or any of the hollow organs. And then you can compare that to the patients that do not have are not receiving antihypertensive therapy, and you can see that the event rates are similar. There is no benefit according to this data, and again, this is not a clinical trial. This is just looking at claims data. But based on this, you cannot make the assessment that there is a benefit when it comes to number of events that are VEDS related between patients that are on antihypertensive therapy versus non-antihypertensive therapy. Again, this underpins the reason why it is important to do appropriate clinical research and why the Discover trial was started. I have these slides in the middle thinking that it would be a bit more interactive when we are live on stage, but since this is a virtual meeting, please keep your questions to the end, and please share them in the chat box, and Ryan will make sure that questions will be answered at the end of the meeting. Very quickly about the VEDS study. Most of you know about this. There's a multi-center investigator-initiated randomized controlled open-label blinded assessment phase 3 study, which was performed mostly in France with a few sites in Belgium. Initiated in 2003, so it's 21 years old. So there's Celiprolol has clearly been investigated for quite a long time. And this was the first time that there was a real scientific assessment of the use of Celiprolol. Obviously, we've learned a lot since then. In those days, you wanted to be a little bit more careful when you put patients on what then was perceived a pure beta blocker. So the increase in the dose was quite slow. So every 6 months, a dose increase, which means it will take a year and a half or 18 months to get to the optimal dose. And then the primary endpoint was decided to be the time to first occurrence to a primary cardiac or arterial event. They plan to enroll 100 patients and plan to follow these patients after the enrollment of the last patient for about 5 years. Genetic testing in 2003 was not widely available, or if available, fairly expensive. So they used the Villefranche criteria, which are the clinical criteria for the diagnosis of vascular Ehlers-Danlos loss. Once the study was ongoing and after the study was finished, all these patients actually did receive a COL3A1 genetic test. So we do know the subcut of patients that actually were diagnosed that way. So if you look at how it was designed, you start out at baseline. Once you met the inclusion and exclusion criteria, and I said the Villefranche criteria really have to do with it being quite often an event in the family or a personal event. And then if the mutation is known, then you obviously fulfill the criteria, and then some minor criteria. And it is one of the major criteria. There's a minimum of 3 of the major criteria, 2 of the major criteria. Or if you don't have a major criteria, 4 of the minor criteria. Some exclusion criteria based on treatment and pregnancy that you would see. And then once the patients were dosed to the optimal dose, they would be followed for up to 5 years. The study was stopped early at 53 patients, so they did not meet the 100 patients. Now, why do people choose a number to get to an endpoint? Why would you need to enroll 100 patients? Why not 50? That has to do with a statistical analysis plan. There is a way to calculate benefit, and you have an assumed effect size. And based on that, you want to make sure that what you find is a true finding. So you set it to a p-value. And based on that, you can do a power calculation and you get to 100 patients, right? So 53 patients, basically, it makes it more difficult to make an appropriate assessment whether or not the finding is a true finding or a chance finding. The trial was stopped early, though, because it was such overwhelming benefit. As you can see here on the right-hand side, primary endpoint. In the blue line, we see patients on Celiprolol, and on the red line, the patients that were on control or on placebo. In this case, patients were not given placebo; they were just not treated with anything. So look at survival rates, right? So 100% is, you mean 100% do not have an event. And once you go down, if you get to 0, it means that all patients would have an event. As you can see, at 48 months, which is 4 years in, the blue line basically plateaus at 20%. So patients on Celiprolol over that time period had only 20% of patients had an event, where patients that were not on Celiprolol, 50% of those patients had an event. You can look at how you calculate that. There's a p-value, or how statistically significant is this, or how accurate is this data. Anything under 0.05 is considered statistically significant. And here we see a p-value of 0.04, which in a patient population of 53 is a nice number. With that, it becomes unethical to continue the study and not provide patients that are not on Celiprolol to withhold Celiprolol for that. It is so clear that the drug works based on this specific data set. And because of that, this is why the trial was halted. It was considered unethical to continue the trial if there was clear evidence that Celiprolol had such a beneficial effect. So as you can see, primary endpoint, just arterial dissections, significant difference in chance of having an event, 20% versus 50%, with a hazard ratio of 0.36, which is a p-value of 0.04, which means it's statistically significant and clinically meaningful. If you add in the primary and the secondary endpoint, you find exactly the same. The p-value actually becomes more pronounced because the number of patients that had a secondary event in the control group was higher, so 24 versus 61%. Small hazard ratio with a p-value of 0.01, clearly clinically meaningful and statistically significant. As said, most patients post hoc, meaning after the trial was started, did receive a COL3A1 genetic test. If you just pull out those patients that had a positive COL3A1, so 33 out of the 53 had a positive test, again, you see a statistically significant improvement. And I will show you it in the next slide. So this is the COL3A1 subpopulation, primary endpoint and secondary endpoint. You can see clearly that at 4 years, there is a improvement in those patients who are on celiprolol compared to control. Actually, if you look at how quickly that happened in the VEDS trial within 18 months. So within 18 months, 60%, or within 18 months, 60% of the VEDS event had occurred in this group. So there's already a statistically significant impact. So you don't have to treat that long to see an effect. You can see the split between the blue line and the red line occur very quickly, and at 18 months, that difference is statistically significant and clinically meaningful. These patients and a second cohort of patients were followed in an open label extension study, or actually a long-term observation study is what I should say, where patients were followed between 2000 and 2017. So these are patients that were both in the VEDS study, but also patients that did not participate in the VEDS study, but were either treated with celiprolol off-label, as a physician wanted them, or did not receive celiprolol. And here you can see the long-term duration of this. So this is not in months, this is follow-up in years. And you can see that benefit goes out years, and this is proportion of patients still living. So we don't look at events. This is survival rates, and you can see a significant survival benefit for those that are treated. The top one is the pure survival rate. The B one at the bottom, that is actually looking the difference between if you're treated with 400 milligram versus less than 400 milligram. As you can imagine, we keep on saying it's not a beta blocker, but it is an alpha agonist. So it does cause vasodilation, which can cause lower blood pressure in some patients. Some patients just do not feel happy with that. And therefore, you lower the dose to 300 milligrams. So the blue line is the patients on 400 milligram, and the red line, those patients on less than 400 milligram. It is not a big difference. It is statistically significant. So it's clear that the higher dose derives the best benefit in this specific cohort that was followed for up to 17 years. A lot of people often don't talk about is celiprolol does have side effects. And so these are side effects that were found in the VEDS trial specifically. And we can see that dizziness in 10% of the patients, they do report dizziness, abnormal tiredness, headache. These are, events related to blood pressure, most likely. And you could also argue that tendency to fall may fall into that. But we can see that there are specific side effects with celiprolol that do cause patients to either hold treatment with it and choose an alternative. So it has to be clear that 10% of patients will have side effects when using celiprolol. Okay, last stretch. So the Discover trial. So we started this trial in 2022. As said, the reason for this was that the FDA provided what is known as a complete response letter, which is a nice way of saying the FDA did not approve your drug. Please go away. They usually say, please go away, but do the following. And what they requested us to do is to run a well-controlled, well-designed clinical trial that would show clearly from a statistical and clinical perspective that celiprolol had a significant benefit and a clinically meaningful benefit over placebo. Usually you're supposed to run 2 of those trials, and usually there's a lot of unclarity as to how to run these trials. In this case, the FDA was so impressed with the VEDS data that they felt that they wanted to give us a helping hand. And that is known as a special protocol agreement, in which the FDA provides additional assistance on how to design the trial. That is great because it helps you run the trial. The not so great is that you're stuck with that agreement and you can't really move away from that. Now, we think, or actually convinced, that we have the optimal trial to run, and we're very happy with the assistance that the FDA provided us in regards to the design of the trial as well as the running of the trial. Before we started, we already brought the top thought leaders from both the US and the United States together to help designing the trial. So we had people from the open label extension study or the long-term follow-up study, as well as the VEDS trial, as well as from the Swedish cohort, as well as experts from the United States to help us design the clinical trial. So the trial is called the DISCOVER trial, the decentralized study of celiprolol on VEDS-related events reduction. It's a phase 3 trial, and the things that we're really looking at is the same as what most trials look at, is time to first occurrence of a confirmed clinical event. And that could be a fatal or non-fatal arterial event, including the sectional ruptures, the uterine rupture, a colon rupture, or any unexplained sudden death. And then we look at the difference between the celiprolol group and the placebo group. Secondary objectives obviously look at incidence rates, or not just the time, but how often does it happen, looking at a variety of additional safety and tolerability issues, as well as changes in quality of life. This trial is a 2-to-1 randomization. We wanted to minimize the number of patients that needed to be exposed to placebo. The trial therefore is a little bit larger, but the total number on placebo is smaller than it would be if you would run a 1-to-1 trial. And we wanted to make sure that we would provide optimal benefit to patients, because we are, based on the VEDS data, convinced that there is a significant benefit for patients with VEDS to be treated with celiprolol. We wanted to minimize the numbers on placebo. We did argue with the FDA to run this as an open label trial, meaning no placebo group, but the FDA was adamant and strict in their assessment that any clinical trial requires a placebo group, and it doesn't matter that you basically withhold appropriate treatment to patients. As part of that discussion, twofold, patients are allowed to have background treatment. So most blood pressure lowering drugs are allowed, ACE inhibitors, ARBs, or calcium channel blockers. Since the FDA considers celiprolol beta blockers are not allowed as concomitant medication in the trial. So that is the only difference that you would see. So you look at how the trial is designed, very similar. First of all, obviously, patients need to be genetically confirmed. So we do not use the Villefranche criteria. We will use COL3A1 genetically confirmed for pathogenic, or likely pathogenic variants, with the exclusion of haploinsufficiency. We are planning to enroll 150 subjects with a 2-to-1 randomization. So for every 2 patients on celiprolol, 1 patient on placebo. And that number was derived from data from the VEDS study. So we looked at the VEDS study specifically for the COL3A1 population, worked our way back for the difference that you see that I just showed in the previous slide, powered it at 90%, assumed a 10% dropout rate, put our p-value at 0.05, and then you come to 150 patients that you need to enroll to be sure to get there. The agreement with the FDA is that we would look at the data after 46 events. So once we reach 46 events, we will look at whether or not the trial is successful. Based on the event rate in the VEDS trial, that would take about 3 to 3.5 years. So we put that at 40 months. And we have also agreed with the FDA that we will be allowed to do an interim analysis at 28 events. That is an important thing to do, because usually when you do an interim analysis, you pay a statistical penalty because of, you basically have 2 shots at goal. That is statistically considered unfair. In this case, this is pre-specified in the step analysis plan, and we do not pay what's known as the alpha penalty on that. So we are allowed to look at it without changing the p-value for the study. Patients that are currently on a beta blocker need to wash out of a beta blocker. As said, you can do that quick or you can do that fast. In the original trial, we were doing that slowly. We have a trial amendment, so that actually goes a little bit faster. Secondly, we are up titrating a lot faster. So the dose increase in the VEDS trial was every 6 months, and here we do it every month. We have quite a few patients enrolled that are on the maximum dose of 400 milligram. We have not seen adverse events associated with that, and no dropouts associated with that. And all patients currently are on the 400 milligram dose. So that actually worked quite well. In regards to the washout of the beta blockers, we've changed the language in that regard, as well as some of the inclusion criteria after discussions with Dr. Dietz. So we now are obviously concerned for patients that have asthma or have diabetes, in which case beta blockers may have significant downstream effects. We want to make sure that that isn't the case. And then also we made sure that patients were not without beta blockers for too long. So the time between the washout and the start of the double-blind placebo-controlled trial is also significantly shortened. That's it. This is what the trial looks like. We are some way down our recruitment and are waiting for events. So once events happen, inclusion and exclusion criteria. Sorry, I'm going too fast. Inclusion criteria, this is fairly straightforward. Written informed consent, willingness to obtain a magnetic resonance angiogram or an MRA at baseline, need to be COL3A1 positive, 15 years of age and older, and able and willing to discontinue beta blockers 28 days prior to enrollment. Exclusion criteria, the usual. If you don't have any of these, if you had a qualifying event or what would be a VEDS-related event within 6 months prior to screening, if you're unable to discontinue beta blockers, then a whole list of additional cardiovascular issues that may be impacted by the treatment of celiprolol, as well as severe renal impairment. Clearance of celiprolol obviously is important, and as well as hepatic or liver impairment. In regards to the discontinuation of beta blockers, it is not written in the protocol, but our discussion with the principal investigator, as well as the study staff, is if a patient is on a beta blocker for any reason outside for the treatment of vascular Ehlers-Danlos, such as unstable angina or hypertension, malignant tachycardia, we suggest the patient not to hold the beta blocker. If you're only on a beta blocker for the treatment of VEDS, please stop. Otherwise, we prefer you not to stop, although it is a discussion between the patient, the principal investigator, and the treating physician to make that assessment. But we would certainly not try to push you into this, because we do not want to have additional adverse events because you will halt the beta blocker and then transfer to placebo. So once the study is up and running, which it is, and events happen, and we've had events occur, these need to be looked at. So there's an independent adjudication committee. Dr. Braverman, Dr. Bork, and Dr. Boutier are 3 VEDS experts to look at that. So once an event occurs that is being assessed as possibly related to VEDS, they receive a package with all that information, and then they make an assessment whether or not this event qualifies as an event as defined in the protocol. If that is the case, the patient is then taken out of the study, given the option to continue in the open label extension study, or if the patient prefers to withdraw, they're also allowed to withdraw because they've met the minimum time to be in the study. Once 5 events have occurred, we have a data safety monitoring board that looks at the data. They look at the data in an unblinded fashion, so they know whether the patient is on placebo or not on placebo. And that is really to make sure that, and I'm going to give you an example, say there's 20 events, and all 20 events occurred in the placebo group and 0 events in the celiprolol group. This is purely hypothetical. That is obviously not what's going to happen. But for example, in that case, you would argue that there is such overwhelming evidence for the efficacy of celiprolol that you do not want to expose patients to placebo, and you would then halt the trial. It could also go the other way around, obviously. So that's what the data safety monitoring board is looking at regular intervals. So as said, the DSMB meets every 6 months, starting after 5 qualifying events. We have not reached 5 qualifying events, although we did have qualifying events. And I said, potential for allowing to halt the study early based on overwhelming efficacy or futility as determined by the DSMB. And then the adjudication committee, they review the potential events as they occur, and they have done so quite well. Last thing I would like to impress on you, this is a decentralized trial. So this is not a regular clinical trial where patients go to their physician, who is part of the clinical trial setup, sign consent at site, and then go back for regular visits at site to do the assessments. And the trial is managed by their physician that treats VEDS. In this case, since most VEDS patients actually, have regular contact with a VEDS care physician, there's not much that you can do, right? There's not a lab test you can do to follow you. You can do an MRA, or you can do an angiogram, or an echo maybe. But unfortunately, the disease presents itself by an event. So in that case, all we are looking at is if you give a patient Celiprolol or placebo, when does the event occur? So in that sense, once a patient signs up on the trial, they sign up themselves through a website. We contact the treating physicians. We discuss this with the treating physician and exchange medical records. And then the patient enrolls. The patient enrolls at home. They get a healthcare or a home visit by a healthcare professional that does the enrollment, does the blood draw that you need to do in the beginning for the genetic testing, collects urine, because we do urine sampling to ensure patients that we know what the pharmacokinetics of Celiprolol is. And otherwise, the patient gets the drug at home, takes the drug. They need to account for the drug. So there's an app on their phone or an iPad in which they can do that. That's it. So we have basically a patient-centered setup. No need to visit anyone. We will come visit you, or we will call you. So there's an overarching CRO, which is a contract research organization, which is basically a company that manages the whole process. We have a super site called Science 37, which is a company that is extremely experienced in running decentralized trials. So they have the principal investigator, they have the study nurses, so they will do all that. And then some other companies that do the lab and the central labs, some bioanalytical lab, genetic testing, and then depots to send out the supplies and distribution of everything. And then obviously the adjudication and the monitoring board. That is it. And the trial is therefore, once the patient signs on, it runs itself, is what we say. It's not that easy, unfortunately. But once the patient is diagnosed appropriately and enters into the trial, our experience, and as I said, we've been up and running for almost 2 years now. We have had no issues once the patient is going through this system. And with issue, I mean, is no complaints that patients feel that it's too cumbersome or too complex or too difficult to follow what they need to do. This clearly is a setup that is convenient for patients. So the recruitment process. So once you screen, it takes a bit of time to get through that. So we start the trial once you sign consent and start your if you're on a beta blocker, once you start going off the beta blocker, or once you start up titrating the Celiprolol placebo. Takes about anywhere between 4 to 7 weeks. If you're not on a beta blocker, between 9 and 60 weeks if you're on a beta blocker. So basically, the moment you sign on to the website versus getting your first dose, between 2 and 3 months, roughly. And that we have experience in quite a few patients that have gone through that process. So you sign up, then we have to request medical records. Takes a bit of time to get that. They need to be considered. They have to be read by the principal investigator and the medical staff. Once you see if you qualify, you consent to screen. Then there's a screening procedures. You enroll, then you need to wash out of the beta blockers. There's genetic testing. There's a baseline MRA that needs to be done, that needs to be scheduled. All these things will happen in the background while the patient is just at home. So the patient does not really notice too much about this. But from signing on to actually being fully enrolled in the trial and being dosed, as said, 3 to 4 months. And the last but not least, this is the website. So discoverceliprolol.com. Patients can sign up there. Just click on the interested in learning more button, fill in your detail, and a medical professional from Science 37 will contact you. Very important to know. You will not be enrolled without at least at a minimum being your treating physician being informed, and we usually like to have at least an agreement that the treating physician, so your VEDS primary physician, agrees with you participating in this trial. And with that, I am going to give it back to Ryan and see if we have any questions, and if Dr. Genemetre and I can answer any of these. Thank you very much.
Thank you. Yes, we did have a couple of questions that have come in, and I also am looking forward to just maybe a little bit of candid conversation between the two physicians as well. And so I will pose those questions too, and then keep an eye out for any others that come in. But if we have some time remaining, perhaps I can sit back and enjoy a little bit of chat between the two of you here as well. So the first question is, it looks like it's somebody that had probably applied to be involved in the study previously and has not heard back. And so they wonder if that indicates that they have not met the inclusion criteria.
No, that has to do with the hiatus between Acer basically halting recruitment. The study was never halted, but recruitment into the study was halted, and the financial difficulties that we were in. That recruitment hold from patients being contacted or patients being responded to was up to 14 months. So there is indeed a possibility that patients were not contacted for that period of time. Had nothing to do with them not being qualified. If you're being contacted and you're not qualifying, you will be informed that you're not qualifying.
Very good, thank you. There's another question looking for some clarification that if a patient were to experience a qualifying event, are they removed from the study?
If you experience a qualifying event, that event is being assessed by the independent adjudication committee. If the independent adjudication committee identifies this as a qualifying event, and I'll give you an example. You have a dissection in your renal artery, in your kidney, in your artery to your kidney. Most likely that would be a qualifying event. So MRIs, additional information, surgery that probably follows onto that, whether it's a placement or a stent or a sleeve or other surgery. All that information is sent to the adjudication committee and they will make an assessment. If it is decided that it is a qualifying event, you will be removed from the trial as a participant in the trial, although you will then have the opportunity to enter in the open label extension study, which we will continue following you. You will still be part of the study. The only exception is you will receive Celiprolol, so you will not be on placebo.
In discussing the trial with some community members, it seems that there are some barriers that they are discussing and sharing with one another that are making them a little bit hesitant to consider the study. One of which is that if they are currently on a beta blocker, and especially if they have been on the beta blocker for some time without having any sort of events, you can imagine that there's some fear with regards to discontinuing that medication. So wondering if you have any sort of advice or words of wisdom with regards to enrolling in the study.
Yeah, it's unfortunately this is a placebo-controlled trial, so that is a real risk. If you are stable and have not had an event for a long time, you're stable on cardivolol or any of the other available beta blockers in the United States. It's a personal decision. It's a decision between you and your treating physician, whether or not you are willing to take the risk of being on placebo. We would fully understand that you don't want to do that. That is a reasonable decision. Participating in a clinical trial when appropriate treatment is available, and as part of the trial, appropriate treatment may be withheld, it almost becomes an altruistic thing to do, right? Why would you participate in the trial? Well, that is the clinical trials are run for the greater good of humanity, or on the other hand, to satisfy the regulator to get the drug approved. So it becomes a very personal decision, and as I said, that should be a decision between the patient and their treating physician. What would be the most appropriate way to do that? And we would fully understand if you would say, not for me.
Would a patient have an option of perhaps switching to an ARB or something instead before enrolling?
That is possible. Obviously, then we would like you to be stable on that for, I think, 3 months is the protocol specific requirement for that. We would have no issue with that. That is really up to you. As said, ARBs are allowed as concomitant medication in the trial.
We are aware that there is some mouse data that would suggest perhaps an increased risk while taking celiprolol. There's also mouse data that suggests a benefit. And so I know that that is a barrier to enrolling that some patients are discussing. So if you could address that, please.
Yeah, it's, without trying to be disrespectful to mice, you know, a mouse is not a human being. The reason you do clinical testing in mice is more proof of concept. Can you show that a specific mode of action, you know, if it works? And you usually use genetically modified mice to do so, right? And we are not genetically modified human beings. In that regard, and it's maybe an odd analogy, but you know, most cancers can be treated in mice, right? But that has not translated into you know, enormous advances in cancer treatment in humans. So we've got great advances in cancer treatment in humans, but it's not because we figured out how to do it in mice. That has to do with other scientific advances. So I would take the mouse data with a grain of salt and look at it, what it actually shows. It does not show that we cured the mouse. What it shows is that the mechanism of action and the you know, chemical sequences we look at, you know, do translate as what happens in a mouse. It shows how the drug works. It does not show whether or not it actually prevents a cardiovascular event in humans.
We have just a few minutes left. I wanted to make just one quick closing comment, is that I'm seeing in our foundation chat that a number of physicians are participating in other physicians' webinars, and we're doing the same here. And I think that that's just very encouraging to the community to see that experts are talking to one another and having open conversations about the progress that we're making with regards to treating their conditions. So with that, I'll give you each maybe a couple of minutes just to share some closing thoughts, if you wouldn't mind.
If I may ask a question to Adrian, I think it was a very interesting trial since I think in the methodology, the decentralized trial, I think it's first very interesting. The fact that you have a 2:1 ratio. I have a couple of questions, technical questions. You mentioned that you were monitoring the celiprolol by urine dosage. Is that prospective, or you mean to get that at the end to see per protocol instead of intention to treat analysis if the results will be different?
Well, first of all, that data is collected prospectively, so it's part of the assessments that we do. We collect on 6 different occasions, so at baseline 1 month, 3 months, and then the normal sequences throughout the trial. 24-hour urine that then is analyzed for levels of celiprolol or its metabolite. That obviously stays blinded because that would unblind the patient, unfortunately, if you would have that data earlier. So we will not look at that until the database lock has occurred, and then we can look at that data.
Okay.
It is mostly to do with that celiprolol was approved in 1986. So most of the pharmacokinetic and pharmacodynamic data is relatively old, and we just wanted to make sure that we have at least an assessment of how this works. So it gives us a little bit about renal clearance. It gives us a little bit about, you know, are patients actually taking the drug as they said, and are there significant differences in 24-hour area under the curve pharmacokinetics when assessed to urine.
I had another question, since actually all the events that you are counting are not only arterial events, but also digestive event or uterine event. Are you randomizing the history of previous digestive event, colonic perforation, for example, since obviously it will influence the course of possible recurrency within the trial after that?
Yeah, that is a good question, and actually interesting some of the findings you had in your trial in regards to the difference between the males and females.
Yep.
So we do not stratify. So the only placebo and active group have the same number of patients is age. So we stratify for age. I think the cutoff is above and below 32, but not for previous events or sex or any of the other things that we could stratify for.
Okay. If I may give a very positive appreciation about this trial, I think it's very important to have medical evidence for or a particular treatment. And I think that's the fact that Celiprolol is a particular drug, as you mentioned, Adrian, very well, in terms of its effect on the beta, but also the alpha receptors, is very important to test this effect. My fear is that in your trial, the fact that part of the patients might have other treatment, as you mentioned, might probably more or less, even though it's against placebo, you will test also the effect of particular combinations in terms of efficacy. How did you evaluate this kind of risk in your trial?
So we know that probably, and this is data that is not necessarily verified specifically for the VEDS population. So again, this comes from more recent claims data that we looked at, that roughly between 60 to 80%, depending on where you look, of patients with VEDS are on ARBs. So that a significant number of patients in our trial will be on ARBs. I think that last time I checked, it was just below 70% of the patients. So that has a significant impact on how we analyze the data. Luckily, the trial is large enough, 150 patients. So even if it is, say, 70% of patients are on ARBs, you still have quite a large number of patients that are not on ARB. So you still have an assessment of 40 against 110 patients, which, you know, with the stability that you see, I mean, this is not a hyper heterogeneous disease. It is a heterogeneous disease, but you know, there's limitations on how heterogeneous, meaning time to event this disease is. So we do feel that the data will be stable enough to make that assessment. It would be interesting to see. It may very well be is what you showed in your trial, is that the combination with celiprolol is significantly gives significant benefit. There's a study that was published from, I think it was Glasgow, where they did a large retrospective cohort where they looked at it, although the numbers were a bit off, where you saw the opposite, that ARBs alone were better than ARBs plus celiprolol. You know, if we find that, as a company, we would be very upset, and we do not believe on any data that we've seen so far that would be the case. However, the outcome of the study is a scientific outcome, and whatever is best for patient is best for patients. And that's really what we're trying to achieve here, is ensuring that we have robust, derived data that supports the optimal treatment for patients with VEDS. That's really what we're trying to do.
My very last technical question would be about the blood pressure monitoring. So you mentioned that hypotension was one of the exclusion criteria at the beginning. Did you mean notion of history of symptomatic hypotension, or blood pressure level below a certain threshold at the entry in the protocol? And how are you going to manage this blood pressure during the protocol?
For patients, the first 6 months, they will get a health home visit by a healthcare professional that will do blood pressure measuring at visit. So we have, you know, point in time assessments of blood pressure, and my background is cardiovascular surgery. So when people say they put on a blood pressure cuff and they say I got hypertension, it's like, well, no, your blood pressure was measured in a certain way today, and this is what the number shows. That it's not the same as hypertension. So it is really based on history. So we'll go to the medical records. You know, did they have syncope? Is there dizziness? You know, were they treated for hypotension? Things like that. I mean, and there's obviously, you know, patients with POTS or other specific hypertensive episodes. Obviously would probably be excluded based upon the assessment of the principal investigator. They're not specifically exclusion criteria in the protocol, but it's, you know, it's a medical assessment to ensure that patients are as safe as possible during the running of the trial. So medical history, blood pressure measurement, and then just, you know, measuring adverse events. If a patient is on 400 milligrams of celiprolol and all of a sudden has more dizzy spells than they had ever had, well, then there's two things you can do. You can lower the dose to 300 milligrams, or assume that the study is not for the patient, and withdraw the patient due to adverse events.
Well, thank you again. It's very interesting program.
Well, thank you, Dr. Genemetre, for, first of all, an excellent presentation before us. You perfectly set the scene for this one. And you know, very interested to see for both compounds how that would be beneficial for patients. And I think you know, the whole treatment, treating physicians as well as patients are interested as to what would be the optimal treatment, and whether it's alone, combination, or you know, patient specific as to what may be best. I hope that that both of our studies will provide the answer to the patient population within the next few years.
Well done. Thank you.
Once again, I would like to thank Drs. Courtel and Genemet for joining us today. As a reminder, on Friday, we will re-air both of their webinars. Dr. Genemet's at 3 p.m. Eastern Time, and Dr. Courtel's at 4 p.m. Eastern. This webinar that you have just watched actually will be uploaded to YouTube as soon as possible following the conclusion of the conference as well. If there are any questions that you had that were not addressed during the webinar today, you can submit those to marfan.org/ask to our Help and Resource Center. So once again, thank you so much, doctors, and to the community, thank you so much for joining us today. We appreciate your time.
Thank you very much.